Early detection and the availability of treatments to a large number of these rare diseases is a huge challenge. It's a bit of a chicken and egg situation, since investment in any individual rare disease is non-existent due to the tiny market size. However, when you add up all the rare diseases of the category, it's a total market worth investing in.
Welcome to the Transfer Files, inside the world of federal innovation. This is your insider's look at how federal laboratories bring groundbreaking technologies to market. I'm your host, Andrea Nelson. In every episode, we'll bring you insights and expertise from throughout the world of tech transfer. In the journey of commercializing a technology, bringing something that was created or discovered in a lab and turning it into something that can be sold and distributed, a big part of the task is clearing the hurdles along the way. And there can be hurdles and potential hazards along the whole way. In our last episode, we heard from Michael Salgaller from the Technology Transfer Center at the National Institutes of Health, or NIH, about marketing technologies. But there can be challenges to even getting to that discovery. Today, we're diving into those early stage challenges with two incredible guests from NCATS, the National Center for Advancing Translational Sciences, also at NIH. Amy Gadhia and Balki Balakrishnan join us to talk about how NCATS breaks down the barriers that stand in the way of drug discovery and development. From streamlining innovation to rethinking how we approach drug development, Amy and Balki shed light on how NCATS is helping to reshape the journey of commercialization, right from the starting line. Let's get into it.
First of all, thank you both so much for being here and joining me. And I would love if you could just give a brief introduction to what NCATS is.
Yes, thank you, Andrea. So my name is Balki Balakrishnan, and I would love to introduce how NCATS was established and where we are going. So the National Center for Advancing Translational Science, or NCATS, was established in December 2011, and it's the latest center to be established within NIH. The center is dedicated to systematically reduce barriers that stand in the way of drug discovery and development and remove inefficiencies that slow down any kind of drug development process. So the mission of NCATS is to get more treatments to all people more quickly. The audacious goals include increasing the number of treatments fivefold from the current ability to treat 5% of known diseases to 25%. And to make the process twice as fast as the current pace. So these audacious goals, in order to work on that, NCATS works on these challenges by developing the science of translation, reducing inefficiencies, and developing platform technologies that can be used to make drug discovery and development more efficient. Just as an example, 90% of all drugs that enter into human clinical trials fail. And you might ask why. But if better predictive models can be developed, they can tell scientists ahead of time what molecules won't work in humans. The use of three-dimensional human organoid models, along with stronger biomarkers and more efficient clinical trials, would increase the predictive power and efficiency of drug development. And simultaneously addressing multiple diseases that share common features also leads to additional efficiencies and increases the odds of drug discovery. NCATS has made considerable progress in all these areas, as you will see from this podcast, and we have shared these findings very widely. So I will stop with that introduction, Andrea, and nice to join you again.
Excellent. I thank you for that. And NIH is unique in that it grants funds for external research, and it also conducts research in-house. So can you talk about how that dynamic shapes how tech transfer happens in your office?
Sure, Andrea. This is Amy Gadhia, co-presenter with Balki, my director at NCATS OSA. I'll talk more about my background and how I got into tech transfer later in the podcast. But for now, I'm just going to jump into an answer to your excellent question. So NIH's dual role in the scientific ecosystem as a funder of research through our extramural staff and an active doer of research through intramural labs makes it a uniquely powerful scientific organization. NCATS in particular embodies this dual strength by ensuring that the most appropriate points in the research journey, judicious combinations of both components are brought together synergistically. And one example of this is with the tissue chip program that grew out of a unique partnership between NCATS scientists and outside scientists who received extramural funds. As our program blossomed, NCATS used its convening power to make sure that each advance was carefully validated and taken to the next step. Disease and application experts were invited to the mix so that the entire scientific community of tissue chip developers spoke with a unified sense of purpose, reduced duplicative experiments, and shared resources without having to start from scratch.
Yeah, thank you, Amy. I am going to present as another example of this convening power and how NCATS was able to combine the powers of internal research as well as external grant funding powers. So NCATS is an inventor, funder, and convener. Like I said, can be illustrated through another example, which is the development of 3D organoids or three-dimensional human organ artificially made in the lab and used. So as the field of 3D organoids began to grow, NCATS funded a number of critical studies through the Cooperative Agreement Grant Mechanism, which is a unique grant mechanism that NCATS uses fairly frequently. And this grant mechanism requires intramural scientists at NIH and NCATS to work with NCATS-funded extramural scientists. This mechanism allowed much more flexibility in bringing different disease and organ experts to share common resources and exchange best practices while honing their skills on their own specialized tissue or disease. While 3D organoids and other ex vivo systems cannot fully replace animal experimentation, human organoids have become more and more predictive of drug toxicity and efficacy. These developments may have taken place independent of NCATS, but without the combined firepower of the collaborative science that NCATS fostered through the intramural-extramural collaboration, it would have taken much, much longer. Now, collaboration and cross-disciplinary research are two of the several important values for NCATS. And we believe that wider adoption of these principles can actually speed up scientific research, especially in a complicated field like health science research. The Technology Transfer Office at NCATS fully embraces these principles and has been instrumental in ensuring the success of many of the collaborative and resource-sharing agreements that had to be established and put in place between NCATS and the outside partners in each of these fields described here. So I hope Amy and I have shown you that NCATS is in a very unique place of combining both the internal laboratory capabilities as well as the external partners that we fund through extramural grants.
Absolutely. Would you mind discussing the agreements and the main uses and differences among the various agreement options?
Sure, Andrea, I can chime in here. So I understand we have a broad audience, so my descriptions and explanations will be a broad, big picture, and I won't get too far into the nitty-gritty. However, for anyone who has more specific questions after the podcast, they can feel free to reach out to me. My bio is on the NCATS website, and I'm happy to serve as a resource. With that said, there are a number of different agreements that address varying tech transfer objectives. I think most folks are probably familiar with a confidential disclosure agreement or a CDA, which is typically the gateway agreement to facilitate discussions among scientists so that they may share information beyond that which they've published on. At NCATS, this information may include chemical structures and other places as well as other proprietary information that may be the subject of a filed yet unpublished patent application, for example. And if the scientists want to share tangible materials like cell lines or mouse models, then we'd help negotiate and execute a material transfer agreement or an MTA. And if after initial discussions, the scientists decide that they want to collaborate, then we may enter into a variety of collaboration agreements, including a research collaboration agreement, an RCA, which is probably our most common agreement at NCATS, a cooperative research collaboration agreement, or a CRCA, or a cooperative research and development agreement, a CRADA, which I know many federal agencies use, and which is appropriate if the collaborator will be providing funding. Now, I know I threw a lot of alphabet soup at you there, but the RCA, CRCA, and CRADA are all collaborative agreements which require a research plan. The CRADA includes a roadmap for managing joint IP and licensing. And where joint IP is developed under the RCA or CRCA, then that joint IP might be managed by an interinstitutional agreement, an IAA, another acronym there. The IAA speaks to which party takes the lead in patenting and licensing. So these agreements, which I just mentioned, are the most common agreements. Of course, we do more complicated agreements as well as custom agreements. And if the parties want to outline their working relationship with broad goals to effectuate their understanding, then they may put in place a memorandum of understanding, an MOU, or a letter of intent, an LOI. And where clinical research is conducted, then a clinical research agreement or a CRA is appropriate. All in all, we look at the goals of the collaboration and then decide on which of these agreements can be used alone or in combination.
Thank you for walking us through the agreements and the alphabet. It's a lot, but boiling down those main purposes and goals really helps clarify. And when I think about NCATS and translational science, specifically, there's this inherent focus on applying research, and that seems to align so much with the principle behind tech transfer. So can you talk about how those two intersect?
Yeah, sure. I'd be happy to talk about how they intersect at NCATS in particular. So we have a therapeutic development branch. TDB is the acronym, and it includes bridging interventional development gaps, and that's bridges, and therapeutics for rare and neglected diseases, and that's TREND. So TDB includes bridges and TREND, and these scientists develop IND-ready therapies for consideration by the Food and Drug Administration for clinical testing. And IND stands for an investigational new drug application. NCATS Bridges assist researchers in advancing promising therapeutic agents through late-stage preclinical development toward IND applications and clinical testing. And through the TREND program, NCATS provides collaborators with access to significant in-kind resources and expertise in the preclinical and early clinical development of new therapeutics for rare conditions and neglected tropical diseases. And the office that I work in, the Office of Strategic Alliances, helps these programs with due diligence related to IP, licensing, and we also help determine the best collaboration agreement mechanism. And even after the collaboration is done, we help with amendments, making us a whole lifecycle partner. We work very closely with TDB throughout the collaboration to ensure that the appropriate tech transfer mechanisms are being utilized where necessary. Again, making us this whole lifecycle partner.
What are your favorite commercialization successes that you or your office has had?
Yeah, thank you for the question, Andrea. And thank you, Amy. You described all the collaboration agreements, mechanisms, and the philosophy behind how to develop drugs in a more efficient manner in great detail. So what I'm going to do is I'm going to present one example, and Amy will present another example. Now, just to be clear, NCATS was established in 2011, so we are fairly young, and drug development takes 10, 15 years or even longer. So we are still very early in our pipeline. So commercial stories, in terms of commercial stories, we will talk about two very promising drug development projects which haven't reached the pinnacle of achievement, which is reaching human use, but we are on the way there. In the first example, I will highlight the discovery, development, and successful out-licensing of a new first-in-class cancer treatment molecule, and the molecule is called Metarrestin. And the reason it was named Metarrestin is because, as the name suggests, Metarrestin would control the metastasis of cancer cells through a new and different mechanism. This phenomenon was jointly discovered by NCATS scientists with their academic collaborators. through a phenotypic assay, which means through an observational assay that was capitalized on the fact that metastatic cells had an unusually large number of perinuclear vesicles. In other words, they look different from normal cells. Just outside their cell nucleus, there are a large number of perinuclear vesicles. And using this observation, the NCATS scientists developed a very painstaking imaging-based screening assay through which they discovered the molecule Metarrestin. Now, NCATS has taken the lead to develop these molecules much, much farther than most academics would. Most academic scientists would develop a molecule like this, publish it, and then immediately out-license it. But because we had multiple collaborators, and we had our commitment and resources for the project, and we had multiple internal levels of development, because of all of these reasons, it made sense for NCATS to take the lead and take it through the phase 1 trials. Finally, most recently, the phase 1 asset have been exclusively licensed to Oncala Incorporated for further development. So phase 2 studies and subsequent development of this Metarrestin molecule would hopefully develop into a new molecule for cancer therapy. So we are all eagerly awaiting that. Now, Amy will describe a different story from a different perspective on another development project that we're very proud of.
Yes, thank you, Balki. I'm going to talk about NCATS and Cincinnati Children's, who collaborated to develop a small molecule for treating myelodysplastic syndrome, or MDS, and acute myeloid leukemia, AML. The new treatment will have potential to provide long-term benefits for both MDS and AML by inhibiting IRAK1/4 and FLT3, including all FLT3 resistance mutations. My office worked closely with Cincinnati Children's Innovation Ventures, our counterpart, to explore pathways to support technology development through the late preclinical development phase. Cincinnati Children's Innovation Ventures is known as CCIV. They recruited an experienced entrepreneur in residence to manage the project operation and coordinate product development by NCATS and Cincinnati Children's investigators. CCIV also helped form a new company, Curome Therapeutics, whose mission is dedicated specifically to the preclinical and clinical development of the novel inhibitors. NCATS entered into an interinstitutional agreement. You might remember me talking about that earlier when I was talking about different agreements. And this interinstitutional agreement, or IAA, allowed CCIV to take the lead in filing patent applications, marketing, and exclusively licensing our joint IP for the new inhibitors. NCATS and CCIV worked together to enable Cincinnati Children's to enter into an exclusive license with the startup Curome for the IP, which covered the novel inhibitors, and for the treatment and diagnostic applications for AML, MDS, and solid tumors. Cincinnati Children's, Curome, and NCATS also entered into a CRADA, another agreement that I discussed. And the CRADA provided Curome with an exclusive option to license future IP relevant to the inhibitors. The CRADA was renewed for an additional 3 years because it had been so successful. And CCIV filed and secured patents for the composition of matter and the methods of use for the inhibitors. The exclusive license agreement and the CRADA provide Curome with a sustainable intellectual property portfolio with over 80 patent applications filed. CCIV partnered with CincyTech, a Cincinnati-based seed stage venture capital fund, to facilitate funding for Curome, the startup. Later, Curome closed a Series Seed round and then Series A by an international venture capital, or VC, firm focused on the life sciences sector, and a New York City-based hedge fund. So it secured a good bit of funding. And since Curome was founded, it has used the proceeds from its capital raises to fund ongoing preclinical development in collaboration with investigators here at NCATS and at Cincinnati Children's, thereby dramatically accelerating the drug development. So in summary, a clinical candidate has been identified, GMP manufacturing of phase 1 drug product supply is complete, and an IND was opened by the FDA on February 9th of this year, 2024, within 30 days of submission. This unique three-way collaboration is a productive and effective model for accelerating translational drug development at both the scientific and commercial levels. A nice success story that we'd like to talk about. Thanks, Andrew, for the question.
Absolutely. Thank you both for these great examples of success, and really puts things into concrete terms when it comes to putting all of these elements together. So I'm curious, in the current landscape of the innovation ecosystem, what would you say is the biggest challenge in getting life-improving technologies into the market?
Sure, I can chime in here. I think that one of the biggest challenges is getting interest in early stage technologies, given the risk that's involved. We heard Balki talk about how long drug development takes, and there's a lot of risk. And one of the other big challenges is attracting capital and expertise for these types of technologies. Risk capital in the life sciences has steadily decreased in the last decade as companies have become more risk averse to supporting early stage projects. And the establishment of NCATS was a partial response to this need. By reducing the barrier to the earliest stages of drug discovery and development, NCATS hopes to remove some of these challenges. The other big challenge in the field is to speed up the diagnosis of rare or unusual diseases that can generally take several years to complete. This diagnostic odyssey costs society close to $1 trillion of burden per year when you take the direct cost to the patient and add in the indirect costs of caregivers and loss of productivity. Early detection and the availability of treatments to a large number of these rare diseases is a huge challenge. It's a bit of a chicken and egg situation, since investment in any individual rare disease is non-existent due to the tiny market size. However, when you add up all the rare diseases of the category, it's a total market worth investing in. NCATS is pioneering ideas and experimentation in this paradigm of working on many diseases at once, so that rare diseases become not only tractable, but also become worthy of financial investment. And reducing the risk of biomedical product development by systematically making the process faster and more efficient and developing new paradigms that translate the learning from one case to many can help society get more life-improving technologies to the market faster.
What technologies or areas of research do you expect to be central for your lab in the next few years?
Yeah, thank you, Andrea. I think this is a very important and extremely complicated question. NCATS has been at the forefront of several new technology development platforms. We have chosen to work on projects that will have the impact on the greatest number of diseases. And further, we have concentrated on rare diseases that do not have treatments and don't attract much commercial attention. Like Amy was mentioning, many of these rare diseases have small markets. So from a commercial viewpoint, we have to combine them into one group of diseases so that they're worthy of commercial investment. As an example, under the Bespoke Gene Therapy Consortium, NCATS is leading the way in identifying and overcoming common challenges in the development, manufacture, and licensure of gene therapy treatments. Many of the lessons that we learn are shared. Other innovations that we are currently pioneering include the use of techniques such as single IRB or institutional review boards for distributed clinical trials, innovating in finding and stratifying clinical study participants, and streamlining the recruitment process. All of these are very big challenges, especially in rare disease drug development. We find that the translation of life science discoveries into useful treatments is not only a scientific challenge, but also an engineering and a social challenge, as some of the above examples prove. Hence, the discovery, development, dissemination of any new tool or technology that makes the process of translation smoother and faster will be central to the success of NCATS in the coming years.
Now, I would love to back up and dive a little bit into each of your backgrounds. So could you tell me a little bit about your professional background and how you got into tech transfer?
Sure. Thank you, Andrea. I think I'll begin a little bit way back when I got my PhD. So after I got my PhD, Andrea, I worked in the biotech industry for a number of years. And I worked in a number of different functional areas. Obviously, right after PhD, you start working in research and development. So I started working in R&D. And then because I was working in a small company, I started helping with the manufacturing processes, so moved into manufacturing, and then into new product development, and finally into business development. So this kind of a transition happened somewhat naturally for me because I was working in a startup biotech that had a number of different financial resources as well as a number of different product pipelines. After a while, all the while, I was working in cross-disciplinary teams and interacting with customers as well as collaborators. And even though at that point, I was still working primarily as a scientist, I was doing a number of licensing deals and also getting more and more into collaborations and technology transfer. So in the year 2000, when a pure technology transfer opportunity at NIH came into my direction, I decided to make a full-time commitment and full-time transition into tech transfer, moved to the East Coast, and started working at NIH. So my transition into technology transfer was somewhat slow and organic, but I'm glad I chose to work in technology transfer full-time at NIH. Here at NIH, I can enjoy the best of all the three worlds, science, law, and business, which are all required in order to make a successful technology transfer career. And I also enjoy the fact that NIH has a positive impact on society. And within NIH, I worked in a number of different components: the National Heart, Lung, and Blood Institute, the main office of tech transfer, the FAES, before I joined NCATS in 2011, the year it was established. So that's briefly my journey into tech transfer and where I've been for the last 20 plus years.
Thank you so much. And Amy, I'll give the floor to you.
Sure, thanks. So I had worked on technology transfer policy for international jurisdictions when I was at the International Intellectual Property Institute, which is a think tank focused on promoting IP and transfer of such worldwide. Before that, I had worked at an automotive company and a law firm. And after IIPI, I also worked at a law firm doing mainly patent prosecution, where I had the opportunity to work on a license agreement. I'm trained as a patent attorney, but I thoroughly enjoy negotiating the different aspects of the license agreement type and collaboration agreements. So I focused on that when I was at the law firm. And then I decided to research institutions that did that exclusively, IP licensing, that is. And I came across a university tech transfer office not far from my law firm where I interviewed and landed a job. I worked there for several years in varying roles. And then I joined NCATS, NIH, where I have been since 2016. And the rest, they say, is history.
Beautiful. Thank you both. And now, knowing that background, what insight from your experience most impacts how you approach your work now? As Balki pointed out, it's tech transfer brings together a lot of different disciplines. So go ahead. I'm curious to hear.
Yeah, definitely, Andrea. I think one of the things that I feel fortunate enough to be working in this field is right from the beginning of my career, I realized that no work in the scientific field can be done in a vacuum or by yourself. Even though, people sit in front of laptops and do a lot of things, I realized early on, for example, when I was doing cell culture or complex assays, I realized that none of the work that I did would be possible without the complex web of discovery, material suppliers, infrastructure support, and all the other things that go into a complex experimental field like life sciences. And collaboration and cross-disciplinary work has not only been a key to my success, but also has been the sole source of my job satisfaction. Because these qualities are greatly valued at NCATS, and because these attributes come to me naturally, I feel a sense of pride and satisfaction at work. I also believe that each individual at work is different, and even if they may not share the same values or work style, each person has something unique to offer. And having such an open mindset can be a source of joy, since you're able to learn new things every day from people around you. Lastly, Andrea, I like to be a peacemaker. So when I do negotiations, when I do collaborations, I always look to expand the pie and explore win-win situations. And this trait also has served me well and made me enjoy the field of technology transfer.
I actually share many of the same thoughts as Balki in terms of win-win. My prior experience at a think tank, a law firm representing companies and government agencies, and a prominent university tech transfer office all helped me understand collaborators' interests. So I've sat at all the different seats at the table. And because of that, I think that I can better understand their interests. I manage an IP portfolio that's joint with NCATS as well as outside entities, mainly universities. And again, I think that my extensive university experience allows me to understand how to negotiate with them and, like Balki, arrive at win-win solutions. We're always looking to grow the pie, which I think is particularly helpful. And with the complex collaborations and in general, that way everybody wins and we all further our incredibly important missions of improving public health.
Wonderful. And I'm going to go really broad here. In your opinions, what is the biggest challenge facing the federal tech transfer community today, and how do you deal with it?
Sure. I'm going to talk about technology marketing in particular, and I think it's challenging for the tech transfer community as a whole, especially in this age of information overload and multiple possible channels by which individuals get information. Targeted marketing through personal contact still remains the most effective means of finding potential partners for your technology. But this is very labor-intensive and time-consuming. We use a combination of technology-specific webinars and deeper scientific discussions among scientists following a CDA to generate the next level of interest. Ultimately, tech transfer is a contact sport, and it will require substantial levels of time commitment to build and maintain relationships. We also find that the scientists themselves play a major role in identifying potential partners. At NCATS, we as the tech transfer professionals are fortunate to have a very close working relationship with our scientific workforce. We are co-located in the same working space and overlap heavily in the organizational chart. We engage in regular and open communications. Despite all of these enabling mechanisms, though, technology marketing still remains a big challenge.
So Andrea, I'm going to talk about another big challenge that I think faces the technology transfer community as a whole. And that challenge is finding talent. Now, you might say, there are a lot of talented people in this world. Why is it so hard? And the reason that technology transfer is uniquely finds itself in a challenging spot is because the field of technology transfer requires a knowledge of law, business, and science or engineering. So, even finding individuals at two of these areas of expertise is challenging. Another big challenge within the field of technology transfer is the fact that this particular profession is fairly mobile. Because of the unique talents, the individuals can take their talent to multiple different organizations, and the desirability of these skills in various types of organizations makes the retention and the attraction of technology transfer professionals to your organization to be a fairly big challenge. As such, organizations have to work very hard to motivate and retain these skilled individuals. Now, on the supply side, NCATS is fortunate to be part of a larger ecosystem where technology transfer individuals are hired and trained. That is all of NIH. At NIH, we put a lot of effort in training our workforce and providing growth opportunities for them. And NCATS is particularly well suited for that kind of an exercise. Our organization, NCATS, is built on collaborations, and hence the leadership itself of NCATS values our office. As such, technology transfer staff at NCATS feel highly respected and valued. Also, the larger NIH technology transfer ecosystem provides our office staff with many networking and mutual learning opportunities. The NIH tech transfer staff meet very regularly in a variety of situations, which in turn provide each of the members a sense of larger community. I think lastly, the public health mission of NIH, and in particular, the NCATS mission of finding more treatments for all people faster, serves as a great motivational tool. I think all of these combined make NCATS and NIH to be a wonderful place for technology transfer professionals. But again, finding the right individuals. Attracting them, keeping them motivated is a big challenge in the field of technology transfer. So I'm going to stop there and see if Amy has additional thoughts to add to this staff challenge.
Yeah, I can chime in here and attest to this challenge, as I was involved in recruiting and hiring at the University Tech Transfer Office where I worked. Thankfully, there, as here with the NIH, we have an incredible mission and stellar reputation, which is a big draw for professionals. But still, the combination of the educational background and the right work experience is a challenge to find, regardless of all of that. But I think that FLC and organizations like it do help as they provide training and convene professionals together, and together we grow the field. So thank you for that, Andrea and others who are involved.
Oh, well, thank you both so much for both of your time and your insights. I think a lot of the things that are underlying those challenges you talked about are the same reasons that make it really valuable to have these kinds of conversations, although tech transfer probably happens a little differently in every office and maybe measured, its success is measured a little differently depending on the office and the agency and the context. But hearing about how it works at NCATS and at NIH and the similarities and differences, I think is going to be really valuable to a lot of our listeners. So thank you both again so much. And is there anything else that we have not touched on that you would like to bring up?
Thank you, Andrea. I think you covered a lot of ground here. I especially want to thank FLC and Autumn for all the wonderful work they do in fostering a sense of community for technology transfer professionals like us. We make a career out of it, we make a living out of it, and we enjoy working with all the tech transfer professionals who are nurtured and motivated through the FLC's work as well as Autumn's work. And we are very happy to share whatever experiences we have, whatever challenges we have met, and how we have solved them. So Amy and I look forward to a continued conversation on these topics. And thank you, Andrea, for taking the time to really sit with us and have a discussion. Thank you again.
Thank you, Andrea. I echo Balki's sentiments and look forward to continued conversations. Take care.
For more information on Amy, Balki, NCATS, and anything else we've talked about, check out the resources linked on this episode page at federallabs.org. Thanks to our producer and digital marketing manager, Carly Dunow, graphics manager, Danny Batchelor, web content and systems manager, Jess Klein, and the FLC staff and community for your support. Thanks for listening. We'll see you back in 2 weeks on Transfer Files Tuesday. Thanks for tuning in to the Transfer Files, your insider's look at federal tech transfer. Don't forget to like, subscribe, and stay connected for more episodes exploring how hundreds of government laboratories and countless industry partners are driving innovations that shape our lives.